This video will focus on the structure of DNA and RNA. Our learning objective for this video is to be able to identify the basic structures of DNA and RNA and be able to label their components. So let's begin with an overview. DNA and RNA are nucleic acids, one of the four main classes of macromolecules. Nucleic acids have several levels of complexity. The smallest level of complexity is the nucleotide. These form repeating units called a strand, and these strands are bound together to form a double helix. Now, our DNA double helix will further condense in multiple steps into a chromosome, but this will be the focus of a later video. So our basic structure of a nucleotide is made up of three components. The first here is a phosphate group. Second is the sugar. We have two different sugars, deoxyribose in DNA, ribose in RNA. And then we have five bases, four in DNA, adenine, thymine, guanine, and cytosine, and four in RNA, adenine, uracil, guanine, and cytosine. These three components are linked together to form our nucleotide. Our phosphate group is bound to our sugar, as is our base. Notice the numbers on our sugar. These numbers are important. These refer to the carbons that are in the sugar. And how we term them, this is our 5' carbon. So our 5' carbon of the sugar is bound to the phosphate group, and our 1' carbon is bound to the base. This is true for both RNA and DNA. These nucleotides are linked through phosphodiester linkages. In a phosphodiester linkage, the phosphate group of one nucleotide binds to the 3' carbon of another. And this is the way that multiple nucleotides become linked in a strand. This linkage is what gives our DNA molecule directionality. You can see here in our diagram we have 5' to 3' here. This refers to the direction as you proceed down the molecule and that you will encounter the carbons on your sugar, 5' to 3'. This is going to be very important later when we talk about subjects like DNA replication and transcription. The phosphates and the sugar form the backbone, while the bases project inward. These bases are going to interact to form the helical structure. Here we see our complementary base binding, adenine to thymine via 2 hydrogen bonds, and cytosine to guanine via 3 hydrogen bonds. The two strands bind to each other in an anti-parallel fashion. And what this means is that one strand, 5' here at the top, 3' here at the bottom, the other strand that's bound to it is going to run in the opposite direction, 3' here at the top, 5' here at the end. These two strands are then twisted around a common axis to form a helix. It's a right-handed helix, so as you're looking down the molecule, it would turn clockwise. And if you're still looking down that molecule, you would see about 10 bases per complete twist of the molecule. And this is all generated by that complementary base pairing. This is a hard and fast rule of DNA structure. Adenine always binds to thymine, cytosine always binds to guanine. The basic structure of RNA is largely the same. Two main differences, uracil instead of thymine and ribose instead of deoxyribose sugar. Typically, our RNA molecules are much shorter than our DNA, and usually they are single-stranded, though they will form some short segments of double-stranded. Our RNA is typically copied from our DNA via transcription. And RNA structure is really important to a lot of the work that's done in our cells. The RNA molecules will form these short double-stranded regions, and these are usually uh, for forming a secondary structure, right? As seen in this 16S rRNA molecule, R being ribosomal RNA. So this molecule is going to be really important for translation. Similarly with this one down here, tRNA, also really important in translation. Its ability to perform its duties in translation is tied to its secondary structure, the way that the single strand folds back on itself and interacts through complementary base binding.